And the next speaker is Ángel Merchan from the Cajal Cortical Circuits Lab, directed by Javier de Felipe. So Ángel obtained his PhD in telecommunications engineering. Then he did a postdoc at Harvard Medical School. He later joined the group of de Felipe, and now now he's working at the Center for Biomedical Technology of the Universidad Politécnica de Madrid. And he has been working on the study of the structure of the cerebral cortex of the rat, mouse, and human. So, Ángel, if you are ready, you can start now. We can't hear you. I think you need to turn on your microphone.
Now you should hear
Yes.
me.
Yes.
Okay, I'm trying to share my here. Okay. Bueno, this should be at the beginning. It's at the end. Well, first of all, I'm not a doctor in telecommunications. I'm a doctor in medicine,
Okay.
so I have no idea of mechanical engineering or physics, and formulae, mathematical formula, etc. A few years ago, we were interested in knowing how the movements of blood vessels in the brain would change the behavior or would interact with synapses, perivascular synapses, which are located around this vascular network. Okay, as you know, blood vessels pulsate, but we were not interested in this kind of movement. As you go to thinner and thinner ramifications of vessels, this pulsation is dampened and other kind of movement takes protagonism. And this movement is the possibility of contraction and dilatation of the blood vessel. And this is made with the aim of controlling the blood flow to different regions of the brain. As I said, our interest was to know if these vessel movements could affect the synapses that are located around the vessel wall. Since, as I said, we had no idea of how to approach this mechanical problem, we got in contact with Miguel Urrecha and Ignacio Romero, who are material engineers, and we decided to build a model on this. This image is blood vessels, and in the brain, we have removed everything around them. We have removed any neural tissue that is around these vessels, but please pay attention to the scale because these are very, very small blood vessels. As I said, these are the vessels whose movements we were interested on. Well, this should advance, but it doesn't. Okay, so we had to model the blood vessel and its surrounding tissue. And the idea was to select a region of the brain where blood vessels are characteristically very active with respect to their movements, and to simulate or to build a model of the tissue around. This is the same image as before, and just think we selected a region like this one or this one with a very small vessel across 10 microns thick, okay? And around the vessel, this larger cylinder of tissue. and we are going to simulate the movements in this very simplified system. We first needed to know how vessels move, and we went to the literature and found that, well, blood vessels react to almost anything which is around them in the brain. In this particular example, you have several kinds of inhibitory neurons with the neurotransmitters they release, and they all affect the blood vessels, either for contraction or for dilatation, okay? And don't forget glial cells. Glial cells are not neurons, but they also very closely interact with the blood vessel wall through these specialized structures that we call glial end feet. They cover the outside of the blood vessels, and they communicate and interact with the vessel wall through this specialized end foot. Okay, here, of course, you will have many other neurons. Excitatory neurons have not been represented in this particular example, but their neurotransmitters also affect the vessel movements. Okay, how these substances, how these signals affect the vessel movement? Well, they change. They evoke contractions or dilations in the vessel that evoke local changes of diameter, local changes of caliber. Here you have a couple of samples from the literature in which we can see a dilatation of the blood vessels or a local, very local contraction of the blood vessels. It depends on the stimuli that dilatation or contraction are stronger, or they spread a longer distance in the blood vessel. Okay, as you see, changes in diameter are variable in percentage. So here we have another example, different substances, different stimuli, procedures, and the diameter change in this example is much higher than in the previous one. So we assumed a compromise solution, and we made our vessel to contract or dilate to a maximum or to a 20% of its original or base caliber, base diameter. At rest, they will have 10 microns of diameter, and variation of this diameter would be plus minus 20%. Okay. Next, we have to simulate the perivascular tissue in the brain with special attention to synapses. And we pay the special attention to synapses because these are, as you know, the places where neurons communicate with other neurons, okay? And there, in these particular places, is where these many substances are released. These many substances that can interact with other neurons, of course, and also with the vessel wall. So synapses were our main interest when we performed this simulation of the vessel walls. To do that, we based the location of synapses was based in what we know from three-dimensional electron microscopy. Basically, three-dimensional electron microscopy is based in serial sections from which we make a reconstruction, and in this case, you're going to see how we reconstruct the places where neurons connect synapses within this volume of tissue, which is reconstructed from serial sections. these very, perhaps you don't recognize them as synapses because only the point of contact has been represented, and we can reduce or simplify or idealize the system even more if we consider just the centroids or the coordinates where these synapses are located. Please remember that we haven't reconstructed the whole synapse, I mean the axons, the dendrite, etc., but only the point of contact. In green, excitatory synapses, in red, inhibitory synapses. And these centroids are the positions in space of these synapses. Of course, this follows a rule, and I can tell you that synapses in the brain, at least in the cortex, in the neocortex, are located almost at random. So if you follow this model, if in a model you want to construct, you put synapses more or less at random, they will reproduce what happens in real tissue. Of course, we were interested in what happened around blood vessels. You can here see a blood vessel, and we are reconstructed the blood vessel and the perivascular synapses with the same method as before. And you will see that very similarly, as you've seen before, synapses are located everywhere, everywhere, randomly or almost randomly distributed around the blood vessel too. Okay, green synapses are excitatory, red synapses are inhibitory, much less frequent in the cortex. Well, if we put everything together, we made, I repeat, with the help of these Miguel Urrecha and Ignacio Romero, the engineers, they made this finite element model in which we have represented here half cylinder of the model, and you see here the blood vessel crossing the tissue, and we have represented here perivascular synapses with the same color code as you've seen before. In this case, please remember that we haven't represented all synapses that are located around the vessel. We have only represented 1/10 of the whole amount of synapses that surround a common blood vessel in the brain. What we are going to do is simulate a local contraction and dilation in this central region of the model, and then we will measure. I hope you can see it. I'll repeat it in again. Contraction, dilatation, contraction, and dilation. Okay, and as you see, synapses around the blood vessel move too, because, as you know, the brain tissue is very, very soft. Okay, we can measure in our model the maximum displacements of the surrounding synapses in the model, and you can see here how the maximum displacement is located here, okay, right at the level where the deformation of the vessel wall occurs. And in this case, we have simulated symmetrical deformation for contraction and for dilation, okay? It's interesting to note that the deformation of the surrounding tissue is quite local. I mean, it doesn't spread to the whole model. It only takes place very close to the moving vessel wall, okay? It only takes place close to the vessel wall. And this is interesting. We will discuss later why. We can measure these displacements. And as you can see here, these are different time points, okay? If we concentrate in the maximum displacement, okay, it's 1 micron, but it decays to 0.5 microns, 5 microns from the vessel walls, okay? So whatever happens at the vessel wall, it decays to 1/2, 5 microns far from the vessel walls. And it decays again to another half, 12, 15 microns farther, and so on. So the closest you are from the wall, the most sensitive to these movements are the positions of synapses. This can be represented also by the distance increment between synapses. This is important because if synapses are far from each other, the neurotransmitter that they release always spills over around in the vicinity, in the neighborhood of the active synapse. But if they are close to other synapses, they could crosstalk. I mean, this is a phenomenon that is very interesting because in this case, synapses would not act as individual contacts, but somehow would interact between them, and they act as a collective entity. Okay, so if synapses are far enough and we compress them and they close them and we put them close enough to crosstalk, we are changing the way the cortex is behaving. Okay, but again, this phenomenon is only restricted, let's say, to 5 microns surrounding the vessel wall. So this can be represented as a field of displacements. And as you see, imagine the blood vessel is here. Okay, the vessel wall is here. Remember, only this part of the vessel wall will move in response to a contraction or dilatation stimuli, and this is the tissue surrounding the vessel wall. As you see, the deformation of the perivascular tissue, and of course the changes in distance between synapses is restricted to a very small region around the vessel wall. Okay, we can represent this dynamically with the same color code. You see synapses moving in this region, but not here, or here. Okay, contraction, dilatation, and the perivascular tissue is only affected locally. So, in conclusion, blood vessel movement change the position of synapse close to the vessel wall. Okay, this movement causes local changes in the density of perivascular synapses. The density increases when the vessel dilates because it compresses the tissue around it, and the density of synapses decreases when the vessel contracts, because it expands the tissue around it. Okay, these changes may help to spread or restrict the diffusion of perivascular released signals. Okay, as I said, if sinuses are forced to stay together, more closely located than before, the neurotransmitter that is spilled over around them will crosstalk with other synapses, and also will be able to interact directly with the vessel wall. Okay, so here we have a possible mechanism of interaction or regulation of the vascular activity. Okay, and of course, don't forget glial and fict. They could also be sensitive to those signals and mediate vascular effect. Okay, not only the signals, I mean the neurotransmitters which are released around the blood vessel may act on its wall, but also may act on these vascular glial end feet, which are very precisely located in the most deformable region of the perivascular tissue. Okay, if this interaction occurs, it would affect end feet first. Okay, and they can mediate, of course, glial cells can mediate and regulate and compensate or enhance the effects of contraction or dilatation. Okay, so we have here several possibilities of interacting with the vessel wall just because they move, and these movements, remember, are made to control local blood flow. So local blood flow can be regulated by synaptic activity, either directly or by the mediation of glial cells. Okay, please remember, which I forgot to say, that vascular changes happen in a scale of seconds or even minutes, while electrical activity of neurons and synaptic activity uses a very different scale. It happens in milliseconds. So synapses have time enough to interact with the vessel wall during these vessel wall movements. So this was published some years ago. If you are interested in it, you can find it easily. And well, thank you very much. If you have any question.
Thank you, Ángel, for a nice talk. Well, in